My colleague Remus Draskatis, who was going to chair this session, is having a few problems logging in at the moment. So I'm Lee Rees, I'm the CEO of Aurox, and to get things started and then try and bring Remus in the background. So without further ado, I'd like to introduce our first speaker, Professor Claire Brown, the Director of the Advanced Bioimaging Facility at McGill University. Claire's research lab is studying cell migration, and I think I'm just going to hand over to Claire then. Thank you, Claire.
Great, thank you very much.
Can you hear me?
Yeah, everybody can hear me?
Okay.
Good?
Yeah, I think so. So your slides should be ready to go, Claire. I'll just turn off my audio and leave you to it for a minute.
Okay.
Best of luck.
Great. So welcome, everyone. It's nice to see so many people. I see there's over 400 here at the session, so that's probably one of the larger audience I've spoken to. I'm very excited today to tell you about some work that we recently published in Journal of Cell Science on live cell imaging and optimizing conditions to minimize phototoxicity. And I wanted to give a shout out to Journal of Cell Science for this feature that they have now called First Person, where they highlight first authors on different articles in the journal. And I was very happy to see my PhD student, Alexander Kipas, highlighted. And he did essentially all the work that I'm going to be presenting today. And it's a very nice feature where they interview the students and get a little sense of their research background and their career goals and so on. So I recommend you take a look. They feature several authors each month. So I'm going to tell you today about this project where we were looking at phototoxicity and live cell imaging. And we focus mostly on wide field fluorescence microscopy because I think this is really a tool that's underused and really, really valuable for doing live cell experiments. It's relatively fast and efficient, and it can answer a lot of biological questions, I guess in a simpler way than more complex instruments like laser scanning confocals. So we use it as a main tool in my lab where we study cell migration, and we do single cell tracking experiments. And we're doing this in the context of just understanding basic cell migration as a principle, and also in disease progression and in things like breast cancer. So this is an example here of a time-lapse video. You're seeing snapshots where we're exploring the effect of the power of the incident light versus the exposure time of the camera or of the sample. And the general idea we had, and I had always kind of had a gut feeling that it was better to use lower intensity of light for a longer exposure time than it was to do short, quick exposures with a higher power. And we had never had data to actually show that this was really the case. So that was how this project started out. And what we did is we kept the power multiplied by the exposure time constant throughout the experiments, and we found what we expected, which was that the cell migration slowed down as we went to higher powers and shorter exposure times. So on the bottom panel there, you can see rows plots of tracks, and each track is a single cell. And you can see on the far right side, the tracks are very long and the cells are migrating quite far. This is over a period of about 3 hours. Whereas if you look at the far left side, you can see that the cell migration is really inhibited. So we had presented this work at many conferences. We were working on writing up the manuscript. And our general conclusion was that when you have higher photon density at these higher powers, you're getting more phototoxicity, or more fluorophores are breaking down and creating toxic radicals that are damaging to the live sample. And that was our basic explanation for this process. However, through a rigorous review process, which was challenging but ended up greatly improving our work, we realized that this was actually not the case, and that this was not what was happening. So we stepped back, and if you look at the steps in a time-lapse experiment, if you start by opening the shutter, then you turn on the light source, you expose your fluorescence sample to the light, you collect an image on the camera of the fluorescent signal, you close the shutter, and then you save the image. Then you repeat that at your next time interval. And the key steps are opening and closing the shutter and turning on the light source. And what we determined through the process of exploring this data in more detail was that all shuttering is not the same. So a mechanical shutter, which you need for light sources such as mercury HBO, xenon lamps, or metal halide lamps, are actually fairly slow. And then if you have LED light sources, there are different ways to control that. You can use USB control from the acquisition software, or you can use what's called TTL triggering from the camera, where the camera will trigger the light source to turn on when it's ready to collect light. And we wanted to explore these shuttering mechanisms in more detail to explore a principle called illumination overhead. So we started looking at the physical shutter first. So in the top panel there, you can see the ramp on and the ramp off as the shutter opens and closes. And this is a gradual increase in power as the shutter opens, because it doesn't open instantly. It gradually gets larger and larger. And same for the ramp off. And we used an instrument called an oscilloscope, which you can put right on the microscope, and it will measure the light hitting the sensor. And so you can see here it was taking about 10 milliseconds to open the shutter and 10 milliseconds to close the shutter. And the total scale on the graph is from 0 to 25 milliseconds. We then went to look at the LED light sources, which can be triggered much more quickly using electronics. And here we have USB and TTL options. So the USB, as I mentioned before, can be controlled directly by the acquisition software, whereas the TTL is a cable that goes directly from the camera to the light source. And in this case, the ramp on and the ramp off is extremely fast. The full scale on those graphs is 1.2 milliseconds, and the ramp on and the ramp off time are now about 0.4 milliseconds. So this is much, much faster than a physical shutter. So are there additional delays in the shuttering? So we basically open the shutter, turn on the light source, and then collect our image. Is that the period of time that the sample is exposed to light? So for the TTL triggering, the triggering goes directly from the camera to the light source. So the camera says, I'm ready to collect an image, sends a signal to the light source. The TTL triggering turns on the light source in less than a millisecond. And then we collect our 24 millisecond image. And the signal we see on the oscilloscope is shown there as the black curve. So that the width of that curve is about 24 milliseconds. So the sample is exposed to light for the same amount of time that the sample is expected to be illuminated based on our experimental setup. However, what we found on our system was that when we had the USB cable connected to the light source at the same time as we had the TTL triggering, there was a software delay that was introduced. And even though we set up the image acquisition in exactly the same way, the light was actually on for 137 milliseconds. So this is almost 5 times the exposure time that we were anticipating. So it turns out that there must be a constant of some sort where maybe the images are being saved in the background and the software is saving the image and then closing the shutter or turning off the LED light source. And for the mechanical shutter, this delay is even longer. So we have the longer period of opening and closing the physical shutter, but the software delay is also longer. So we get a total time of 230 milliseconds, or almost 10 times our exposure time. So how does this come back to our experiment? So basically, if we go back to the experiment that I showed you at the beginning, where we thought we were giving basically a constant amount of light to the sample, and we were just changing the power and the exposure time to keep the total number of photons hitting the sample constant. This is indeed not the case because of the illumination overhead. So if you look at the highest power, where we have the 24 millisecond exposure, we're actually exposing the sample to 4 times, almost 5 times as much light. Whereas when we go to lower light power and longer exposure time, the relative contribution of that delay is much, much smaller. So we're only getting 11% additional exposure due to that delay. And this is the case where we're comparing with the USB connection, where we have the 113 millisecond delay. So if we go back to our cell migration curves and we repeat the experiment, but now we actually disable the USB cable and connect the TTL directly to the light source, then you actually see that when we have this higher power of light and short exposure time, we actually see that the cells migrate just fine, and that the decrease in cell migration we were seeing was actually due to more light exposure on the sample. One of the downsides of this is when we disconnect the USB cable, we lose control of the light source from the software. So you can't control the power, or if you have a multi-LED system, you can't control the different LEDs at different times. So this would be a limitation of setting the system up this way. So we started exploring other ideas. So what if you can't avoid this illumination overhead? What if you're using a solid state light source and the bulb can't be turned on and off electronically? So one of the options is to upgrade to an LED-based light source. And we published a few papers and did a lot of work on LED light sources for a while, looking at it from both the scientific perspective of having really good light source stability with the LED-based systems, and also the aspect of getting mercury out of your lab and out of your waste stream. And we were actually able to get funding from McGill from a sustainability initiative in order to replace all of the light sources in our facility. And it has many benefits of removing the mercury. There's no more bulbs to change. The light source is more stable, and you can trigger the light source and avoid illumination overhead. I had a marketing student actually do this cute little YouTube video. So if anybody's interested in looking at that, it's about 2 minutes long and kind of summarizes the benefits. And if anybody's looking to get sustainability funding in this area, I'd be happy to talk offline and give some advice. So what if you can avoid the illumination overhead? So say that you have these light sources and you don't have a budget right now to buy an LED light source. You can upgrade to a higher speed mechanical shutter. So that's a possibility. There are many different versions of shutters that have different speeds. And then if that's also not an option, you don't have finances to upgrade your shutter, then what we recommend is this diffuse light delivery, or using this low light power over longer exposure times. And as I mentioned in a few slides ago, what this does is it just minimizes the relative contribution of this delay, because the delay tends to be relatively short relative to longer exposure times. And we actually go as far as recommending using the entire delay time between imaging to collect light. So the example I show here is a small protrusive area of a CHOK1 cell, and it's expressing a protein pexilin EGFP in focal adhesions. And you can see where the white arrow is, the cell is protruding and new adhesion structures are forming. We were taking images every 5 seconds, which was a good match for this type of kinetics. And we were using the entire exposure time for that. And so we were able to go down to a laser power of 16 microwatts at a 10x lens. And you can see that we get nice high contrast, good signal to noise images, and we're able to follow that process. This is continuous imaging for 20 minutes with the light on the entire time. So if you want to do that, focal adhesion dynamics are relatively slow. So a 5-second delay is quite a long time to collect light. But what if your processes are faster and you don't want to have artifacts in your image? We came up with a protocol, and it's explained in quite a bit of detail in the publication, of how to determine the maximum exposure time for dynamic processes. And basically what we recommend is imaging at high speed, so a speed that you think is much faster than the process you're interested in, and then doing maximum intensity projections at different time intervals to create pseudo longer exposure images. So for instance, you could take frame 1 and 2, frame 3 and 4, frame 5 and 6, and look at maximum intensity projections of those to mimic double the exposure time. You could do the same thing for 3 frames, so 1, 2, and 3, 4, 5, and 6, and so on, and mimic what you would have gotten for 3 times the exposure time, and so on. So the panel on the right shows LLC PK1 cells expressing EB3M emerald. So this is a microtubule tip binding protein that has very rapid dynamics. And then you can see the simulated images below of a region of interest. And at the very bottom, you see just a different lookup table so that you can really see the structures quite clearly. And as you can see, as you go from 10 millisecond exposure up to a 5-second exposure, a 5-second exposure is much too long for this process because now you see streaking, and the EB3 spots now look like elongated stripes in the image. So how can you determine the best time for your exposure time? So we measured a number of different morphological properties of these EB3 spots, and I'm just giving you one example here, which was the roundness. So how round are these spots? And in general, the spots are a little bit oblong, even at the highest exposure time, but you can measure that for many EB3 spots for the different exposure times. So this experiment was done on 5 cells, so each color represents data from a single cell, and using this technique of making these images of pseudo long exposure time. And you can look at where basically the curve starts to decay, or the roundness starts to decrease, showing that the structures are becoming more elongated. And you could say an exposure time for this process of about 400 milliseconds is probably as long as you can go in order to measure this process without having image artifacts. So once you, if you have this ability to go to more diffuse light delivery and use longer exposure times, you also still want to make sure that this process is not phototoxic to your sample. And there's a huge amount of literature out there on this, on how to measure phototoxicity. And there's really nice examples with cell division, with small organisms. And I think the main thing we wanted to contribute here was giving people methods for measuring it a bit more quickly. So a lot of those methods you have to wait, if you're looking at proliferation, you have to wait hours or days in order to make sure that the proliferation rates have not been affected. And the methods I'm going to mention here are ones that can be measured as an endpoint assay, or they can be sampled periodically throughout a live cell experiment. So this is an example of using a reactive oxygen species dye, Aurox deep red, which is measuring mitochondrial ROS. And ROS is a common sort of surrogate for phototoxicity. If there's a lot of toxicity in the system, then the ROS levels go up. We looked at this over the different conditions, and here we're using the TTL triggering to make sure that our exposure times are what we expect. And if you look at the second panel there, you'll see that the ROS production is the same regardless of whether we use the higher power and low exposure times or lower power and longer exposure times. So this is consistent with the cells moving at the same speed and not slowing down. But if we increase the exposure time, which means we're increasing the dose of light or the power exposed on the sample, then of course we do see an increase in ROS because we're increasing the amount of light that the sample sees. And on the right side, what you'll see is the dye before we start imaging. And then on the bottom panel, you see what the cell looks like after we've taken 400 images of pexilin EGFP. And we've done this consistently across, sorry, I don't have the number there, for about 5 minutes, I guess. And at a fairly high power at 21.3 milliwatts. And you can see that the ROS levels are quite high after that significant amount of light exposure. So this is one way to quickly measure toxicity, and we usually recommend to use this as an endpoint assay since the dye is pretty much turned off when there's no ROS production, rather than trying to image it throughout the experiment, because just putting these dyes in and image them can also cause additional phototoxic effects, and also having the dye in the cell could affect the cellular physiology. So another simple example that we've often used just as a visual is we know from our cell migration and adhesion experiments that if we put too much light on the system, the cells start to retract and round up. So in this example, we've just looked at the effect of the illumination overhead when the USB cable is connected over time, and we see that in the normal, non-phototoxic conditions, the cell size and shape doesn't change a significant amount. But in the lower panel where we have the higher light power and significant illumination overhead, you can see that the cell area gets quite a bit smaller as we image over time. And And the last one that I'll mention is mitochondrial morphology or area. So mitochondria are also very sensitive to phototoxicity and ROS production through their morphology. So this is using the MitoTracker red dye, and in the top panel you can see the mitochondrial structure over time, and it continues to be sort of this long spaghetti-like structure. It's very dynamic and moving. And in the lower panel, when we have the illumination overhead and the higher light power, you can see that the mitochondria quickly condense and form little spots around the perinuclear region of the cell. And in the top panel, we were able to image for a full hour, 4 hours with the light on constantly, whereas in the bottom panel, we image only for not even 3 hours before the cells are really compromised. So another caution when using any of these type of dyes in live cell imaging, again, the MitoTracker is kind of a nice endpoint assay, so you can just make sure that the system that you've looked at is your conditions are good. If you do want to follow it throughout, what we do is we'll just image it, maybe every 10 minutes just to make sure everything still looks good, rather than trying to image it every time we take a time-lapse image. And then we carefully modify protocols as well. So for example, I have here on the left the manufacturer's protocol for MitoTracker Red, and then on the right the modified protocol. So we were able to use the one of the lowest concentrations they recommend for the MitoTracker Red dye in this situation. We reduced our incubation time to just 10 minutes, which is a little bit outside the recommended range. And we also wash the cells afterwards to make sure we get rid of any free dye in the media before placing it back in the media. And with these conditions, we've been able to image cells for several hours without any obvious toxicity due to the dye itself. So we also wanted to see if this was a general issue. We had first looked at wide field microscopy, but we also have a spinning disc confocal. So we also use the oscilloscope measurement on that system. And this system uses solid state lasers. So again, they're a source that can be turned on and off electronically. And here you can see these are just offsets so you can see them, but basically the on time and the off time is very fast at about half a millisecond. However, when we started looking in more detail using the acquisition software, we found that there was a significant software delay. So we looked at two different types of acquisitions. When the first one is stream acquisition, where you just open any physical shutters, you turn the laser on, and you just collect images as quickly as possible. Usually those images are streamed directly to the hard drive or the RAM, and they're not stored on the hard drive during the acquisition. And we still found that under that condition, we had a 17 millisecond delay. And then if we set up interval acquisition, so even if we said we want to collect an image as often as possible, so with no set time delay between them, but it would collect an image and then save it and then collect the next image and save it, then we found that we had the interval delay, which was 142 milliseconds. And so it should be 42 milliseconds for the actual illumination overhead. So it's not something that's unique to wide field microscopy per se. So we tried the diffuse light delivery conditions on the spinning disk. So we put our frame rate, our laser power down quite low. And I think I'm missing it on here. I apologize. But we collected 13 slices in a Z-stack with 50 millisecond frame rates. And we collected those over a period of 5.5 minutes, so continuous image acquisition for 5 minutes. And what you can see on the right of the panel here is the upper panel is 3D rendering, just showing the structure of the mitochondrial network. And in the bottom panel, that's the same sample, same cell, after it's been imaged for 5.5 minutes under these diffuse light delivery conditions. So that's what I had to show you today. There's a lot more details in the paper, so I encourage you, if you're interested, to take a look. But right now we're working on a protocol paper on how to measure the illumination overhead. So I know using an oscilloscope wasn't something common that we were using in our labs, so we're writing up a protocol on how to do that. And we're measuring the software and hardware delays on multiple microscopes from different manufacturers to get a sense of the magnitude of the problem and where the problem needs to be addressed. We're publishing a review in Microscope light sources, so we're going to try to really give people a sense of what's out there on the market and what are the pros and cons of the different options. And then as today, we're going to try to raise awareness in the community. I know we were aware that illumination overhead was an issue, but I definitely didn't appreciate the magnitude of the issue. And when we had set up our microscope, we thought we had set it up for the TTL triggering, but it turned out that this USB connection to the microscope was actually causing this additional delay that we were unaware of. So we also want to encourage people to use this diffuse light delivery method to minimize illumination overhead if they can't replace their light source or their shuttering. And we want to encourage the microscope companies also to measure and be aware of what software delays might be present in their systems. So basically, to summarize, if possible, you should upgrade to an LED or solid-state light source and use direct TTL triggering. The best solution is to eliminate this illumination overhead. But if you do have a bulb-based system, then you can use low light power and longer exposure time or diffuse light delivery in order to do this. And you can determine the longest exposure time you can use with the methods I was showing for image analysis and so on. And then once you've figured out the conditions you want to use, then you can measure the phototoxicity just to ensure that your experimental protocol is ideal, looking at cell tracking, cell shape, redox sensitive dyes, or mitochondrial morphology. So I'd like to acknowledge the Brown Lab. So Alex did the majority of this work, but there was help from other members of the lab. The Advanced Bioimaging Facility, so all the experiments were performed in the facility. And then the funding agencies, you can see here, and our collaborators. And I thank you for your attention, and I'm happy to take a few questions.
Thank you, Claire. I apologize for changing head appearance now.
No problem.
I finally managed to log in, so we'll relieve Lee. We have quite a discussion, actually not so much questions, but so discussion going in the chat panel here, mostly around TTL versus USB and all the intricacies therein. But I would like to ask you a more sort of optical question first, if I may. So you clearly advocate the approach, stretch the exposure time as much as you can and reduce the light level as much as you can. But generally, there are kind of two schools of thoughts about photobleaching, phototoxicity. One is saying that only the total dose is important and how you deliver that dose doesn't really matter. And the other school or approach is to avoid high intensities at all costs, just to reduce the intensity as much as you can and stretch the exposure time. Do you subscribe to any of those two opinions, or?
So I think originally I would have definitely been of the latter, and I think that was why we started these experiments, because I was sure that lower light over a longer period of time was much better for the sample, and we wanted to provide the data to show that was the case. I think the example I always think about is getting a sunburn. And if you're out in the sun for 10 minutes in the middle of the day, you get a sunburn, but if you're out around dusk, you can stay out for hours and you don't. And I think that was always kind of the way I was thinking about it. After sort of discovering this illumination overhead and the impact it was having here, I think it really depends on the density of the light. And I think this is going to be more of an issue for things like laser scanning, confocal, multiphoton, where you have a very, very physically restricted dose of photons as well. But in the wide field illumination, I think the light is diffuse enough that it's not, causing a lot of higher order processes to happen.
Yeah, so that kind of gets me to the next following question, is the basic confocal. And there are many different ways to do confocal these days, or confocal or sectioned microscopy these days. And there are sort of wide field techniques, there are quasi-wide field techniques where light is clearly much more uniformly distributed in space and time, as opposed to point scanning confocal or even nonlinear point scanning confocal like two-photon, multi-photon. And super resolution techniques, STED is also guilty of high concentrations of light. So how would you comment that? And is there a benefit in your view to move towards more wide field techniques or more distributed techniques?
So I think I've always been a big proponent of wide field, and as I mentioned at the beginning, I think it's really underused. I think it's a great tool for a lot of biological questions. Of course, knowing the limitations of sectioning and sample thickness and all those sort of things. So I would say as a general rule, I think the wide field based techniques, and I think spinning disk light sheet is another one that really comes to bear, that it's a more distributed light and a camera based technique are good for live cell imaging. I think laser scanning and STED and all these things can be optimized, but I think we need to do the experiments to really find the right regime and the best regime to minimize toxicity.
I guess also it depends on how user-friendly that is made by the manufacturers, because point scanning confocal, it's very easy to overindulge you're short of signal to noise, you just ramp up laser power.
Sure. Yeah. And I think that's often the first, when an image is dim, it's often the first response is to put the laser power up. And we would advocate for, line averaging, slow down. So again, it depends on your biological process you're trying to measure. But I think the other thing we came to conclusion from this study is it's very complex. I even think if we upgraded our operating system, that the delay time might slow down if I had a faster Windows or a faster CPU or GPU. And so I really think it needs to be done with the companies, with the manufacturers, and everybody needs to kind of look at this. But to me, I think the biggest thing is awareness, because we thought we had our microscope set up properly, and we didn't realize that this USB cable was overriding everything.
Yes, and it's always the first shock by the user who sees the sample disappearing in front of his eye.
Exactly.
Claire and Remus, I should just mention we've still got about another 8 or 9 minutes left for questions, but there is a growing list there if you'd like to dip in and answer a few.
I have to admit, I was avoiding reading the chat, so I was talking, so I didn't get so distracted. But
no problem. Some of them do stem right back from the very beginning. I think they're relatively easy questions to start with to answer. You might want to dive in early on.
Those are always my favorite.
Yeah, just to get you going. I think there was one from Jennifer Coolhay. Did you compare the shuttering of different LEDs with each other? That was right back at the beginning of your...
Yep, that's great, thanks. No, we didn't. We did all of this using, these were done with the LED 120 from Excellitus. But that is our plan right now when we get back in the lab eventually. To look at different LED light sources and even the same LED light sources on different software platforms and really try to dig into that and see if it's highly variable or are there certain constants. Like the electronic shuttering should be the same for a given light source regardless of the operating system. But these software delays, I think you'll find will vary from acquisition software platforms.
If I just were allowed a comment about the lasers, you mentioned lasers there, solid-state lasers that apparently can be shuttered electronically, but it's actually not the same thing as LED, where LEDs, you control the current directly, and that can be done with some skill, can be done really quickly. Solid-state lasers are usually very stabilized in a very complex manner. There are a lot of internal feedback loops. So even if you think that you're applying a really precise TTL pulse, what happens internally may be a very different story. So in terms of shuttering, at least in our experience, they're not nearly as good as LEDs, not nearly as pliable as LEDs.
Okay.
So another factor to keep in mind on top of everything else that you already said. So it's
That
so
actually,
Warren problems.
that actually bridges in. I see a question here from Ian Doby of whether the 0.4 millisecond rise and fall time is actually a property of our oscilloscope sensor. And that
light sensor, more like it, yes.
Yeah, whether that's the limit, the time limit of that sensor, and it's very possible because I think the number we got for the lasers was almost identical. So I could look into that. I don't know off the top of my head the time sensitivity of the sensor.
Okay. Anything else that you see easily?
So there was a question, the software we use. So it was the software that came with the oscilloscope. So it took a bit of playing around with it. I'd have to look up the details, but my graduate student was quite good at setting it up so we could measure it directly into the computer and have the actual output data. It's a little tricky depending on the time window you choose to measure. The data points get averaged over different periods. So there's a comment of maximum intensity projection versus summation of the images for the pseudo exposure time experiment. So I guess for the purpose of really simulating a longer exposure time, that's correct that a summation would be better. But I guess we wanted to make sure that we really identified the objects precisely, which is why we chose the maximum intensity projection.
Okay, so
Yeah,
I think we've exhausted our time.
Excellent.
And probably questions as well.
Yep,
so
no problem.
Thank you very much again.
People can always reach out to me if they have other questions.
Yeah, is that okay, Claire? We'll take all the
sure.
unanswered questions from the chat, and we'll send you them via email with people's contact details. If you'd like to, would that be okay? That you could get in contact with them. Great.
Sure. Yeah. Great. All right. Thank you very much.
Okay. Thank you. Okay. Do we have our next speaker online?
Yes. If you can hear me.
Yes, I can hear you. Okay. I
Perfect.
can see you. We can see you.
There we go. Yeah.
Okay. So our next talk this afternoon is by Simon Bennett from Miltenyi Biotech, big and up and coming company of all things biological and now optical. Simon has got a PhD in neuroscience, but I understand that he spent most of his life working for different companies in various roles, and now he's been with Miltenyi quite some time, and he's going to present a talk about the new instrument that Miltenyi has just released to the market, and the instrument that, as far as I can see, takes the concept of multi-channel to its extreme.
Yeah,
we're talking about 100 channels as opposed to 3 or 4 to us mere mortals. So please go ahead and tell us all about it.
Sure. So first, probably an introduction to Miltenyi, as probably not a lot of people have heard of it in the microscopy field. So Miltenyi is a company started magnetic cell separation, primarily concentrating on immunology and things like this. We do a lot of stuff with CAR T cells and things like this. But now Miltenyi's made a bigger inroads into microscopy and things like this. So we've got 2 sort of avenues we're going down, and I'll talk you through the couple of instruments. Mainly we'll concentrate on the Maxima, the one on the left there, but I just wanted to briefly mention the ultramicroscope. And while we're in this session, obviously, for this conference, I would just want to say that we're talking about here fixed imaging, fixed tissue. Now, it makes sense to be talking in this session, but I wanted to make that clear up front that we're not talking about live cells. So the instrument we're mainly going to be talking about today, like I said, is called Maxima, which is, as was mentioned before, a ultra high content imaging system. So we're talking about hundreds of markers from one bit of tissue, one fixed sample, as opposed to lots of things. And the ultramicroscope, which is the other one mentioned there, is about very big cleared samples with light sheet. But yeah, mainly talking about Maxima today. And so the principles of Maxima are mainly around imaging, staining a sample, imaging where that stain is, and then erasing that staining, and doing this over and over and over again to build up your number of markers so that you get huge amounts of data from any sample. This isn't a brand new concept, but the way that Maxima does it in a completely automated fashion, and with these things bringing about from our experience of flow cytometry and from our experience of antibody technology, enable you to do this at a scale that wasn't really possible before in this sorts of level of ease. So I don't know if someone's saying a sound is on and off. Hopefully it sounds all right for most people. Let me know if it isn't. So just to talk about the instrument itself, it's mainly based around an epifluorescence microscope with fully automated sample processing and imaging. So it's a relatively simple device inside, just with lots of components all coming together. What you can see inside there is a bit of the stage that allows you to move the sample around and load the sample in, as well as places to mount certain reagents. So this stage you can do tiling and stitching of your samples and things like this with that. We also have within there the ability to control the fluidics for adding different samples, different antibodies, different solutions on, but that's defined by the experiment that you're doing. And the microscope inside, like I said, is an epifluorescence microscope with a few different objectives. Mainly we're looking at 2 different 20 times objectives and 1 2 times, just to sort that out, just to get a big overview. But these things are a little bit up in the air at the moment and might be changed because the Maxima isn't a fully released system yet. We're still looking for collaborative partners to work on it. So to give people an idea of the sorts of samples you can load in and how you do that, we've got these few different ways that you can put in either a slide of fixed tissue, cultured adherent cells within a plastic insert, or suspension cells on a slide. Now again, the Maxima is going around and staining these samples within these chambers all itself. So what you're putting in is a fixed sample within these different plastics, and then it's taking care of the rest of the experiment for you once you predefine that in the software. We'll go into a bit of the antibody and the staining and the erasing of the sample later on as well. But just to go through this, we've got the imaging frames here for your big slides, your big sections of tissue. The adherent cells, again, you'd have to culture them before putting them, you'd have to culture and then fix them before putting them in the device. And the suspension cells are in this Maxwell slide where you can actually put on a solution of cells in suspension, and they settle into these little tiny divots, and then you can stain them within the Maxima to get huge amounts of information there. So the antibodies is a key part of how this is going to work, obviously. So how we're going to stain our samples, how we're going to image them, how we're going to get our data. Now, I only briefly mentioned before that we're erasing the staining, and this can be done with 2 different mechanisms within Maxima. The first mechanism is photo bleaching of the fluorochromes, so that you're just removing that staining just by destroying the fluorophore, and this is relatively simple. Maxima has to go around with its LEDs and photo bleach each field of view of the 20 times objective, removing the staining there, and then in the next round it's applying antibodies on for different markers and things like this. Now, you have to consider that there might be potential damage to a sample by doing this many rounds of photo bleaching, because we're talking about hundreds of different rounds of this staining. But at our headquarters in Germany, we have done up to 400 markers from one sample using photo bleaching completely. So it seems that's obviously going to be really dependent on what sample you're using and things like this. Another thing to potentially consider here could be steric hindrance of, different antibodies being bound onto all sorts of different markers, but again, that's really, really sample dependent, so it's really hard to say. The other mechanism that Maxima can use to get rid of the staining in the sample is actually a Miltenyi technology called release. This is where we can disrupt an antibody complex, and this actually completely removes the antibodies from the sample, including the Fab fragments that are on there. I mean, this is a whole other technology that Miltenyi use in various other fields like cell sorting and things like this, but it's particularly useful here because we can completely remove that staining all at once, and this is just applying a solution that removes this antibody so that we no longer have to worry about it being bound, and then we can go on and use our stainings, our other rounds of antibody to stain complete different markers. So we've been through the instrument itself, which is applying these antibodies in things like that in a completely automated fashion, and we've got ways of putting our samples in, and we've talked about the antibodies, ways we can disrupt this staining. And then the fourth part, which is almost the most complicated bit, we're talking about the software here. And now, as you can probably imagine, if you're talking about hundreds of markers from any sample, looking at it in just a standard 2D image, once you get beyond 9 overlays, you're starting to lose any idea of what the data could be and how useful it might be. The imaging software that comes with Maxima has been designed really to take full advantage of the data you're collecting from all these hundreds of markers. And a lot of our knowledge from flow cytometry has come into this software. So you can start doing things like gating strategies of cells so that if they're expressing within a hierarchy a certain number of different markers or subsets or things like this, you can then show where they are within your image easier and what they're associated with and things like this. It also has segmentation built into the software, which is really helpful for doing these sorts of analyses. And this is based around the one thing that helps with this segmentation is it uses DAPI every single round to image where the nucleus is, so then it can do these sorts of things. So the software is really important. We're working really hard on that. Again, because this isn't a fully released product, this is still something we're working on a lot at the moment. So there's certain features might change, and we have lots of videos of that software. So again, best to get in contact with Miltenyi local to wherever you are, and people will be, I'm sure, happy to guide you through all these things. So some of the applications that we initially have in mind for Maxima, I mean, there are dozens, but obviously Miltenyi coming from that immunology background, the sort of thing we're thinking about is drug target discovery for things like the immunotherapies that Miltenyi is really heavily involved with. So we're looking at where markers are expressed within certain tissues where we could target that real deep phenotyping of tumors and diseased tissues, so you can get enormous amounts of information from any samples to really guide you through what you want to do. Potentially, I mean, patient stratification could be really interesting ways of looking at this for diagnostic reasons, but again, I think that's probably quite far down the road. So this is a data set that was produced in-house. We've got various markers within this panel, and some of the panels that you can produce for Maxima, I mean, you can produce them completely manually yourself if you want to. You can put in any company's antibodies using that photo bleaching erasing mechanism, or you can create a panel purely out of the Miltenyi releasable ones, and you can chop and change within an experiment. It's completely up to you. But Miltenyi will also be providing predefined panels that you can load into the system so that it can do all of that for you. And then when you scan these things in to the Maxima, it will know which antibodies in which well of the preloaded plate and what dilution to use it at and what sort of staining time and all these sorts of things. But anyway, we've got this panel here where it was done in 35 cycles. So each round of that cycle, we're often applying 3 antibodies as well as that DAPI, so a FITC-P and an APC dye, so that we can, well, fluorophore, so that we can image them with the different LEDs. And these 35 cycles took 35 hours, so it can take quite a while to do an experiment. But again, the amount of data you're getting is huge all at once. So here, an example of where these were, what this panel was used for was looking at CAR T-cell targets in pancreatic cancer. So we're looking at the tumor microenvironment here. We're looking at where the immune cells are, what the tissues are, what's close, what's associated with each other, where we're looking at. And this data set was actually used to find the 15 highest ranked target candidates for a CAR T-cell sort of immunotherapy approach. So then we could then use the knowledge of those markers to design different immunotherapies in the future. And these are the sorts of things that Miltenyi has in mind for these. But again, there's a huge array of applications that are potential here. And here's an example of how you could validate the CAR T-cell lines within the mouse models using Maxima, and understand where different targets might be useful and different ones might not be. So that's Maxima, and we'll go through some of the questions that you guys have. I think that'll probably be the most illuminating thing about this discussions, because a lot of, I just wanted to do a quick introduction to it really. But I wanted to briefly mention the ultramicroscope as well. Again, this is our light sheet microscope for doing enormous cleared samples. The stage can move 1 centimeter by centimeter by centimeter, and some of the microscopes you can actually fit in a whole cleared mouse, for example, so you can image a huge area of a large sample as well, and get very pretty pictures. Again, because we're doing this via the web, we couldn't do some of the videos again for the software of the Maxima, but also for this, so you don't quite get the full effect, but yeah, it's really nice pictures. As a summary, the Maxima imaging platform includes the whole instrument, the disposables, the reagents, the software for fully automated cyclic immunofluorescent analysis with hundreds of antibodies. You can define those panels, or we can provide those panels. It's up to you, but you're getting enormous amounts of data. And this will give you really deep insights into tissue and architecture. The multiplexed imaging allows you to look at subsets of tumors, look at immune infiltration, CAR T-cell targets, and things like this. But then the light sheet we're looking at as well is also involved with CAR Ts, but looking more in 3D specimens and cleared samples and stuff like that. But yeah, that's a really brief introduction to this topic. But yeah, happy to take any questions now.
Thank you, Simon. One question from me first, just to establish some sort of baseline. So you had a slide where you had more than 100 image channels and 35 cycles. So a typical cycle would be 4 stains?
Yeah, so like I mentioned before, we use DAPI every single round for that sort of registration, but then we use different antibodies coupled usually to FITC, P and APC to give you that data, and usually it's 3 per round. It doesn't. That's what we found is the best to avoid any sort of optical crosstalk. Obviously, you can have just 1 per round if you're really concerned about that.
More than 3?
You can design it like that if you want to, but you're going to have to do a lot more careful planning of fluorophores you use and which markers you consider and all these sorts of things.
This 1 hour per cycle time, what is it mostly spent on? It's not on imaging, right?
Well, it depends a lot on how you define what you're doing, because that depends on the field of view you want to image, whether you want to get a big field of view by stitching together lots of images, whether you want to do tiling, it can vary hugely depending on all these characteristics. That's like hour per cycle is just sort of a rough guess based on this experiment, but it can vary a lot. But yeah, like you say, a lot of it's spent on the staining of the samples and then
the preparation for the next cycle, right? And
erasing and all that sorts of stuff. Yeah.
Okay. If you have a quick look at the chat window, which is now enormous, you think you could pick up quickly and answer 1 or 2 questions?
Yeah. So something that a few people have asked that probably is quite important as well is the autofluorescence. Now it takes an image at the beginning of every single round as an autofluorescence subtraction of that sort of thing. If you're working in a really autofluorescent sample, perhaps it's better to stick to the more red LEDs, obviously. And so you'd be more careful about that. But yeah, there is some autofluorescence subtraction going on in there.
And the eraser residue, do you have much problems with those? Residue from previous rounds?
So the photo bleaching style, obviously your antibody's still there. You're just destroying the fluorophore. So there will be some stuff left from previous staining rounds. The release one, we're talking about thin samples here because we're only dealing with epifluorescent microscope, 15 micron maximum, ideally around that or less. So it's relatively easy for the solution to get into the sample, and this releasable solution is it's just a simple enzyme, so it's quite easy to wash on and off. Like I said, we use it in other fields for doing cell sorting and things like that, and from what we've seen there, there's not much or no real residue left over, but there's, again, it's really sample dependent, so it's hard to predict that for every single scenario. But when I mentioned earlier about the 400 markers from one sample, that was all done with photo bleaching, so they got really nice data, even though all those antibodies were obviously still bound onto their markers.
So photo bleaching is clearly a simpler technique to apply, but generally going forwards, do you think release perhaps?
Yeah, so that's our aim, really. We wanted to keep it open, so people, if they've got markers, if they've got antibodies for markers that we don't produce antibodies for, they can still run their experiments, because there's obviously going to be weird and wonderful things people are interested in that we don't make products for. But I think the majority of panels we would see being based around release. They're also quicker, because that photo bleaching has to go around with the field of view of the objective and take the time to photo bleach every single field of view, whereas the release
and it's exponential, it never ends.
Exactly, yeah. So you can put this one solution on and it makes it much simpler. So we're building our portfolio on that massively at the moment, on that releasable side of things, but it's not fully there yet. Okay, well, thank you again. I think we'll move on to the next speaker. Thank you.
Yeah.
Feel free to.
I think, actually, do you think we could just take a few more questions from the chat session? Because I know we've got quite a few people that are asking questions and feeling like they're not getting any answers.
Sure.
Just maybe a few quickly. That would be okay.
Yeah. So, can you use DNA paint? So that's not planned for the first rounds of Maxima. I mean, other people have asked about DNA paint and things like that. The chamber is temperature controlled if that's becomes useful for that, but not for the first round. But we could potentially see advantages of doing sort of DNA paint in the future.
How many color channels can you image simultaneously, was asked?
So it's it's a standard epifluorescence microscope, so you can do you're limited to that. I mean, you've got 6 LEDs and 6 lots of filters in there. So potentially 3 or 4, but again, that's running into more standard problems with normal optical epifluorescence microscopy, and it's designed so that you can just do one color at a time, but 4 colors within each cycle is how we predict it's going to be used mostly.
Okay, quickly, I mean, can you export to Fiji for analysis was a question.
Yes.
No?
So you can do it's that exports TIFF files. So if you want to use other analysis software for analysis, that's totally fine. But it has quite a lot of analysis built into the software as well.
Let's see. Okay, right. I mean, there are quite a lot more questions there. We'll get them across to you.
Happy to take lots of questions
with the contact details. I think there was a particular request for you to contact somebody as well in there. So.
Yeah, we're happy to do that. I mean, Miltenyi are based all over the world, so there may be a local Miltenyi person that may be easier to deal with certain questions, but I'm happy to point those in the right direction as well.
Okay, thank you.
Thank you very much.
Okay. So you probably all met Philippa in the morning sessions, but for those who haven't been there, Philippa has been with Aurox for the last 3 years, has been with Leica before that, and with a few other companies beforehand. And Philippa is going to talk about the latest product from Aurox, but still working in the same principle, spinning this structured elimination microscopy that the company has always championed, and we think we finally got it to a meaningful conclusion. So over to Philippa.
Okay, hello everybody. I feel a little bit humbled to be squeezed in a session with such great talks, but I think it would be unwise of us to miss out on the chance of telling you a little bit about our company and our products. So I'm going to start off by giving you a little bit of history about Aurox. I guess a lot of you may not have heard about our company until last week. We produce laser-free spinning disc confocal microscopy products. We've been around since 2004, so Aurox was a spin-out of the University of Oxford Engineering Department. And we developed equipment such as the OptiGrid, that's what the Zeiss Apotome is based on, essentially. And for about 13 years, we did OEM supply. So we were making equipment for other companies to sell. So these pieces of equipment you might have heard of. So we worked with Zeiss on the Zeiss Vivatome. We designed and manufactured the Andor Revolution DSD range of instruments, so the DSD and the DSD2. And our technology is also inside the Zeiss SmartProof 5 materials analysis product and the 3D Histech confocal slide scanner. So as I said, until about 2017, we were primarily OEM suppliers and our brand was not known by anybody. Then I came on board along with Lee, and our aim was to sell our products to the end user and meet all of the researchers that have been using this technology for so many years. So we instantly rebranded our products, relaunched the Clarity laser-free confocal system, which I'll talk about tomorrow. And most recently, we launched our Unity product, which I'm going to talk to you about in a little while. So firstly, why did we decide to go laser-free? I think you'll see in Tony's keynote lecture in a little while that you do not need a laser to do sectioning, to do confocal microscopy. He actually will show you a really interesting image from a historical slide. So why laser-free? Well, it's cheaper, it's easier to maintain. You don't need expensive service contracts on lasers, which might break down routinely. I know at a lot of conferences I've been to, people are moaning about expensive service contracts and their downtime on their laser scanning confocals. And obviously, if you don't have a laser, that problem goes away. It also makes it a bit safer. So you don't have any laser safety restrictions. You don't need to train your users. Anyone can pick up our devices and use them. Also, without a laser, you're likely to get lower photo bleaching and lower phototoxicity. Although this is a little sample dependent, we do see some different samples behaving differently with different confocal techniques. But in summary, basically it saves you time and money, and it makes confocal microscopy accessible to more people in more locations. You don't need a dedicated core facility to run your confocal. So how do we achieve it? So a lot of people will be aware of the traditional spinning disc systems on the market. So this is your Yokogawa systems, etc. They are all based on a traditional Nipkoff spinning disc. So this is a spinning disc that has pinholes and sometimes micro lenses and other things. Now, Remus and Tony and Mark and my other colleagues, they measured the light throughput of these traditional Nipkoff spinning discs back in 1996, and there was less than 1% light throughput. So they're a bit better now. You get about 5 to 7% light throughput in a traditional spinning disc system. The summary of that is that you need a laser for your illumination. You just need that intensity of light to get the light onto your sample. So at Aurox, some time ago, we developed this structured illumination spinning disc. So instead of very small pinholes, we have quite large letterbox-shaped apertures on this disc. And the grid is spaced as light, dark, light, dark, light, dark around the disc. This means that we can get up to 50% of the instant light through to your sample. This means that we don't need a laser. So you can use an LED light source, or you can use a metal halide lamp. It's up to you. So how do we do this? So we have a white light source. You can see the light bulb on the left of the screen. And as I said, this can be an LED light source, or it can be, hang on a minute, let me just change the transition there. An LED light source or a metal halide lamp, or Tony will show you a little bit later, other light sources too. So we shine the light through our spinning disc, and that projects this structured illumination pattern into the focal plane of your sample. This excites the fluorophores and they emit their light. And some of this emitted light can pass back through the disc. So this is about 50% of the light. We project that light path onto one half of the detector, or in the case of Unity, onto one detector. So this light path contains your confocal section that you're looking for, as well as about 50% of the out of focus light. Okay, so it's kind of a slightly cleaned up wide field image. And then comes the clever part. So the back of our spinning disc is mirrored, it's reflective. So the other 50% of the light that doesn't pass through the disc, is reflected off the back of the disc. We collect this light path as well. So this light contains the rest of the out of focus light. So you end up with two images. On the left, we have in focus, our confocal signal, plus 50% of the out of focus light. On the right-hand side, we have the rest of the out of focus light. Okay. And then what we do is we subtract one from the other, and we end up with our confocal signal. So it's a really elegant way of achieving your confocal sectioning without a laser in a very easy to use manner. So some years ago now, Aurox were thinking about what makes a confocal microscope. Now, I think you'll all agree, anyone that has a laser scanning confocal or a spinning disc confocal, even a wide field microscope, you end up with lots and lots and lots of components. So you have your microscope, you have a confocal device, be that spinning disc or a scan head or something like that. You might want an incubator, you need a detector, a light source, motorized XY stage, you need objectives, you need a computer, and all of this creates a massive problem of lots of cables. You might need an air-conditioned room, you need lots of PowerPoints and things like that. So we thought, well, why don't we just put it all in one box? We want to keep all the advantages of our traditional laser-free confocal systems. So easy to use, low maintenance, affordable, but make it compact so that we can make it even more accessible for researchers. Rita, I'll answer your question a little bit later, and maybe Remus can help as well. So what we did, we developed this all-in-one benchtop laser-free confocal microscope. So it's called Unity. It's just hitting the market now. So it's been a few years in the making, and we're just going into production now. So it's a very exciting time for us. So what's inside the box? So we have all the things that were in that list. So we have the motorized XYZ, we have a laser-free confocal system, we have an integrated detector, integrated light source, integrated objective, and everything that you might need. So just looking a little bit at the light path, so it's a patented original light path inside the system. And as you can see, we have the Aurox disc in the middle, the spinning disc, and the light is coming at the disc from different directions. So we can do a range of different functions with this, and it makes it a very flexible system. So just going down the list on the left, so we have a laser-free system, so no lasers needed and no maintenance contracts, etc. It has an integrated motorized XY stage and motorized Z. So you can do your tile scans, you can do your Z stacks, just like in a normal microscope. The integrated camera inside, in Unity, there are two sensors. There are two 2K by 2K scientific CMOS sensors with 6.5 micron pixels. So it's really optimal for spinning disc confocal, and really sensitive as well. We have an integrated LED light source that sits inside, and also the computer is integrated. So you don't need a separate computer, you don't need a separate monitor, keyboard, mouse, although you can connect a monitor to it if you want to and just use the computer inside. The light path that we have, this complicated light orientation, means that we can look at the sample from above, giving us an overview. So we have an optoelectronic lens there, which gives a 4mm by 4mm field of view of the top of your sample. So this helps you to navigate around, because I think you may have noticed there were no eyepieces on this microscope. So we need some way to navigate around your sample and find your sample. One thing we can do as well is we can run this as a wide field microscope. So instead of doing the subtraction to get our confocal signal, we can just use one of the images, and that is a pseudo wide field image, which is really useful if your samples are difficult to find or difficult to focus on. You can use the wide field mode to find your sample and then switch to confocal mode when we do that subtraction. And it's just one click in the software. Currently, the system will do 3 color channels, and there's a plan to add more in the future. So at the moment, it's Dappy, Fitzy, Tritzy. And one unique feature about Unity is it has one integrated objective lens. You can choose which objective lens is integrated at the beginning. It is possible to change them at a later date, but it's not something you routinely do. So it's all optimized to sit around one lens. I'll answer that quick question. What kind of objective lenses are available? So we're testing a range of lenses at the moment. So for fixed cell specimens or thin tissue specimens where you want to use oil immersion, you could use, say, a 60 times 1.4 NA oil immersion lens, and that will work really nicely. But you can also use all the way down to, say, a 10x lens with a 0.4 NA or something like that if you have thicker specimens that are bigger. So the system is extremely flexible, and you can talk to us about it, and we can advise which objective would be used. And another quick question, the channels acquired sequentially. Yes, they are acquired sequentially. What we do is we strobe the LEDs very fast. So what you get, and if you come to the live demonstration tomorrow evening, then I'll be able to show you how fast that is happening. You almost get a pseudo real-time 3-color image. So it's very, very quick. The lenses, yes, cleaning the lenses, that is one thing to consider. So if you're using oil immersion, we have some approaches to help people to clean the lenses if you're using immersion lenses. Obviously, dry lenses, you don't have a problem. The lenses can be completely removed for deep cleaning, and you can move them up and down in the instrument to access them for routine wiping the lens. But again, we can discuss that with you maybe offline. Okay, just in the interest of time, I'll just crack on a little bit. So we have a Unity control software. As I say, the computer is completely integrated inside Unity. So what we have is we have a tablet interface to control it, or you can control it with a monitor, keyboard, and mouse if you prefer. So this control software is on the tablet interface. It's all touch screen, and it's extremely easy to use. And again, I'll show you tomorrow a little bit of a demonstration of how the software works. But it will allow you to do Z-stacks, time-lapse, tile scans, multi-channel, confocal, and wide field. One thing I didn't mention is that when we're strobing the light source, we only illuminate your sample when you move or when you do something. So when you move the X, Y, or when you move the Z, it will take a snapshot. So it's not illuminating your sample all the time. So we found that sample bleaching in this instrument is very, very, very low, and your sample preservation is really high. And just a very quick question that popped up. When changing the objectives, do you need to change out the Aurox disc? No, not at all. One good thing about our spinning disc technology is that you can essentially use the same spinning disc for a range of different objectives. It's very forgiving technique. So with Unity, you just have one disc with one pattern on it, and you can run all the way from, say, as I say, 10x all the way up to oil immersion, and you don't need to change anything else in the hardware. It's just a toggle inside the software that just tells you which objective you're using, and then it's good to go. So just a quick snapshot of the rear of the instrument. So as I say, we were trying to get rid of this tangle of cables. So the minimum you will need to run this instrument is one power cable, which is just a kettle lead that plugs into the rear of the instrument. And then you can run the instrument from the iPad. If you want to, you can network the instrument, so you can network it to your server for data transfer or for remote controlling it. And we also have USB 3 ports. So if you wanted to attach a monitor and things like that, then that's a possibility as well. We've put plenty of storage on board because obviously, because it's easy to use, we've found that you can ramp up quite a lot of data in quite a short amount of time. So we've put 2 terabytes of image storage on the instrument, and this is upgradable as well. So if you do need more space, you can do that. Oh yes, and Lee has just reminded me that there's 2 connections at the back, which are prepared for you to put your lab CO2 connection in. So I think somebody mentioned about environmental control. So that is coming very soon. We're just finalizing preparations for that, and hopefully we'll have some news about that later this year once the COVID-19 crisis allows us to do a bit more work. Okay. Now, the beauty about this instrument, it's got all that technology packed inside, and this is why it's taken us so long to develop it. It's really small. So it's only about 396 millimeters high, so 40 centimeters high, 22 centimeters wide. It's really, really tiny. It's designed to sit on your laboratory benchtop or in the corner of your core facility and do all your routine confocal work. It's very good for getting very fast imaging, but also really great quality. So as I say, we're going to do a live demonstration tomorrow afternoon where I can show you a little bit of how the system works. It will be a mixture of live and recorded things, because we realized from the iPad we can't visualize it very well using this package. So I've pre-recorded a few scenarios and other things. There's a couple of questions I see coming up about samples. So Jennifer is asking what kind of sample carriers can be used, and Mika was saying, did I explain the sample types? So inside Unity, if I go back to where it's open, we have a number of sample holders. And inside here, if I turn the whiteboard on, it will work. So bear with me. So inside here, in this aperture, is the XY stage. And we have a range of sample holders. So at the moment, we can either have a 2-slide holder, a 2 Petri dish holder, or a 96 well plate holder. So these sample holders they're designed to be removable and cleanable. And then you just insert them into this aperture and they'll be held in place by a couple of magnets. I can show you this again tomorrow. It's a little bit easier on the live instrument. A little bit of a question about vibration control from Chris Wood. Yes, the vibration control is built in. So if we see here where the body of the instrument meets the XY stage platform, this whole top where the sample sits is sat upon some dampeners. And we've done some interesting tests where we are looking at the sample live and we can tap the instrument or tap the desk and the sample really doesn't move. So you don't need to put this on an anti-vibration table or anything like that. I mean, if you're looking at ultra long time lapses and you need your sample to be very, very still and you're looking at minute changes, then I'd say this might not be the instrument for you. But for normal time-lapse experiments, then this dampening control is great. The sample holders are our specific ones. So they're designed to sit in our XY stage, and eventually there will be some identification so that when you put in your sample holder, it will automatically recognize which sample holder you have in place. What is the resolution? Remus may correct me if I'm wrong, but I think we achieve about 0.9 microns Z resolution. But maybe Remus can come online and answer.
Yes, it's actually better than that. But yes, it's in Z. It's definitely very much comparable with the point scanning confocals. Laterally, again, the story is that you get same kind of thing that you get in a confocal. So theoretically, we have a slightly better resolution in the wide field, but in practice, it's about the same as wide field. Mm-hmm. But ultimately, as with always with all these things, it's dependent on objective lens numerical aperture. That's all there is.
Sure. There was another question.
Philippa, early on, there was a question about what happens to the noise when you subtract 2 images. Can we answer that?
Yeah, well, maybe you're the best person to answer that, actually, Remus.
Yes, so I think whoever is asking this question, I can't now find the person, but you're absolutely right. That is a bit of a problem, and that's a price we have to pay for simplicity and speed and everything. There is a little bit of residual noise left after the subtraction. In many cases, that doesn't matter. In some cases, that will be a limiting factor where you can use the system as opposed to, say, point scanning and focal. So, for instance, in very thick and very scattered samples, that becomes a problem.
Sure. I think we did put something about that in our technical notes. So if you go to our website, there's a technical note in the download section explaining a little bit about the shot noise limiting factors in our imaging. What else do we have? Oh, can microfluidics be mounted on the system? That's definitely a plan. Again, we have the ports for the CO2 connections. So if somebody wants to talk to us about microfluidics, then we can definitely look into that. It's not something that's supported just yet, But
I think in this case we'll probably be looking for some sort of partnership. If there is a company that wants to provide that as part of our package, that's perfectly reasonable.
Sure. And then there was one last question that I saw about hardware or light-based autofocusing. No, there isn't autofocusing yet. It's something that we have been looking at, either hardware or software-based, but it's not in this current model. Okay. Oh, and somebody says very thick samples. What's the limit of this technique? So we can use, and my talk tomorrow about the clarity laser-free confocal will have a few more application images in it. You can really, this technique is very forgiving on different types of samples if they're cleared. So any structured illumination techniques and this technique will suffer if there are any aberrations in the system. So if you have refractive index mismatches, things like that, very scattering samples. So we can image very, very thick samples, cleared tissue, things like that, if they're reasonably clear. If it was an uncleared brain tissue slice, then maybe 20 to 30 microns, but cleared mouse tissue, 1 to 2 millimeters. So it really, really depends on your objective and your sample preparation. So, and reducing the aberrations in the system. Okay, I think in the interest of time, probably we should move on to the most of the day. Yeah?
Let's do that. Okay, I can see Tony is already logged in. So our last talk of the day, and it's a keynote presentation, is a personal view of history of confocal microscopy by Tony Wilson. And Tony is clearly the person who is most qualified to give a personal view because he's been there since the very beginning. As many of you will know, a lot of pioneering work in confocal microscopy has been carried out at his lab in Oxford University in the 80s and 90s. And I should also mention that Tony, together with Colin Shepherd, wrote a book with a name that escapes me at the moment. It's called Theory and Practice of Scanning Optical Microscopy, and it's considered a classic and standard reference in the subject. Unfortunately, it's been out of print for a very long time. So whoever can get hold of it, do that now.
Well, thank you.
Yes, Tony, you have a podium.
Thank you. So what I want to try and do in the next half hour is to try and explain how we got interested in what was at that time called scanning optical microscopy, and also to indicate that what people found interesting right at the start is perhaps not quite what people find interesting now. And I first ran a conference in The Hague in 1987, and it's marginally interesting to have a look what those talks were all about. And the other thing I want to emphasize as we go through this is that I started off believing I knew how the confocal microscope worked, and then pretty quickly realized that I didn't really, and it was much more subtle than I had first thought. So it's a purely personal view, and it's really trying to, if you like, sell you old microscopes for new, if you like, children's pantomimes. So you have to be able to at least do as well as people could in the past. And so here are some images I scanned from a book in the Oxford Library. Three-dimensional, very nicely produced bits of I don't quite know what, pollen grain, some sort of bug. And so the issue is, which instrument took these images? And the answer is this one. And these images were, of course, recorded by Robert Hooke in 1665 or thereabouts. Now, fairly clearly, this is not what he saw, but he was able to reconstruct from what he saw these really rather impressive images. So how did we get interested in Oxford? And I think the answer is probably this 1951 paper by J.Z. Young and Frank Roberts, where they started to speak about a thing called the flying spot microscope. And in the first paragraph, they make the remark that surprisingly, very little of the information obtained with microscopes has been quantitative. Most people are happy to sit at the screen and stare at the image, but rarely have people really try to get anything quantitative out of the instrument. They also make the point that by imaging the whole thing in one go, if you like imaging in parallel, that you lose the freedom to tune the resolution, to tune the imaging in a way which you would be able to do if you were to image sequentially, i.e. in a scanning approach. They also make the point that if you have a scanning approach from their flying spot microscope, that the quantum efficiency of the photo detector is much higher than a photographic plate, and hence one could, in principle, use a lower illumination intensity. Their point number 7 was one which got us particularly interested, where they say that the resolution in the ordinary microscope is, well, they use the word halation produced by the interference between neighboring particles, but they claim that in a scanning microscope, since only one resolvable region is illuminated at one go, one ought to be able to improve the spatial resolution. This possibility has not yet been systematically explored experimentally. It now has. So this is really what fired our imagination to start off with. This flying spot microscope, as Rimas indicated, I'm old, so I can remember when television screens had cathode ray tubes to display the image. And what a flying spot microscope is nothing more than a television screen with the electron beam scanning across the phosphor, producing a scanning spot of light, which is then relayed onto the specimen. It scans across the specimen, and you detect point by point, as indicated here, the light transmitted by the specimen, which you then display on a second screen, which is scanned in synchronism with the first. This flying spot microscope was built both in London and in UT Southwestern in the late 1950s. But of course, the problem is this spot is not particularly bright. But fortunately, then came along the laser, and that made a huge, big difference, clearly. So the original motivation was the observation that optics is pretty poor at seeing weak signals on a large background, whereas electronics is very good at seeing weak signals on a large background. So our original thought was, if we can get the signal in an electrical form, we can readily perform contrast enhancement. And here's a fairly early instance where we are just looking at the reflected light on the left-hand side from a rather badly cleaved optical fiber. And on the right-hand side, we are showing that there's a change of about 0.5% in refractivity, which we can map into changes in refractive index. And so we could start off fairly early, getting some meaningful numbers out of the instrument. So we had to do a scanning to get the electronic image. At that time, there were no CCDs or computers. The whole thing was done with analog electronics. And our motivation, really, to be honest, was the enhanced lateral resolution. We didn't really know much about optical sectioning then, and it was this enhanced resolution that was pushing us forward. I know some people have asked about the history of confocal, and I haven't done an exhaustive search, but the first reference I could find was to a paper in 1940 in German in an ophthalmology magazine. Then came a paper in Japanese, which I haven't read for the obvious reason I don't speak Japanese, in 1943. And if you look at the optical layout on the right-hand side, you can sort of see the confocal pinholes. From the same Japanese group in 1951 comes this instrument here. And if you look carefully at the light source in the bottom right hand, you can see a thing marked iris diaphragm. If you then follow the optical path through the objectives upwards and then horizontally towards the photomultiplier tube, you can also see an iris diaphragm there. So this is clearly a confocal geometry. And then, of course, we all know the 1957 patent by Marvin Minsky. So that's a brief history. The first design that we had to find a way to scan. And we scanned using a mechanical scanner. Because there was no way to record the image, we had to have a long persistence television screen, and you could see the image build up as one scanned over the whole of the specimen. And this was the first scanner that we had. It had two vibrator type voice coils scanning a support mechanism with flexible legs, as I show there. This didn't last more than a few days because the flexible legs broke fairly immediately and was replaced with a stretched piano wire system that I show in the image here. And also I show some fairly poor but still early images from the mid-1970s. The optical system you see in the photograph is absolutely awful, but it still worked up to a point. And we got a bit better, and here are some images of a fairly low resolution microcircuit, some resolution test specimen, and the ever-present diatom showing that it actually does work. Interestingly, one of the earlier things people were interested in was color confocal. And here's a conventional image and the confocal equivalent. The early commercial instruments, this is one from Oxford Optoelectronics, which we set up in 1982. This actually had stage scanning. And Texas Instruments was the first customer. We then got into bed with a local company, Du Billier Scientific, which then turned into Laser Sharp and a slightly more professional looking thing. And the screens you see at the bottom of the image are the long persistent screen. This was only in 1984, and then Laser Sharp became Biorad, and the rest is more or less history. So the man behind the work in Oxford was a chap called Rudy Kompfner. He's the man who invented the traveling wave tube. And after his career in Bell Labs, he took 2 jobs. One where he spent the English winter in Stanford, where he worked on acoustic microscopy, and one where he spent the English summers in Oxford, where he started our work on optical microscopy. And here he is. And he was the chap who designed and drew this first scanning stage, which we then slightly modified into the form that you've seen. And here he is with 2 people. And I just draw this because one problem with scientists, no one knows what they look like. The man in the middle is Kompfner. The man at the left-hand side is a chap called John Pierce. And most people have probably never heard of John Pierce, but everyone knows one thing that he did. Bardeen, Brattain, and Shockley won the Nobel Prize for the invention of the transistor, but they didn't name the transistor. He did. So everyone knows his name for the device. No one knows who he is. Pretty much everyone listening to me knows the man on the right, but I'm going to leave it to you to see if you can remember or if you can recognize him. When Kompfner died, Pierce wrote his biography, and he spent a few sentences on his work in Oxford. And he said at the bottom here, he hoped to gain further advantage through observing harmonics of the illumination light. And indeed, that's what happened. Here's the scanning thing again. The thing in the top left is the photomultiplier tube, and indeed these were second harmonic generated images obtained in the late 1970s by Julian Ganaway in Oxford. And the laser was a homemade 1 watt CW neodymium YAG laser. We'd never be allowed to use that now. Amazing that we could do this thing then. A bit later on, we were able to do third harmonic and second harmonic. And here are third harmonic and second harmonic images focusing through a C. elegans nematode. Things have really improved rapidly. Now, 1987, we had this scan imaging technology SPIE meeting in The Hague, and it was very much the start of understanding how these things worked. But most of the talks were industrial. One here, new technique for spatially resolved minority carrier lifetime. Scanning laser imaging of surface chemistry, scanning optical microscopy applied to fluorometry. Again, there were more things, applying the instrument to the measurement of submicron structures, real time scanning by Gordon Keynote from Stanford. And even as back as 1987, there was the inevitable lecture on super resolution. There was biology, of course, but it was very much in its infancy. So optical sectioning is the important thing in the confocal, as we know, and that means you have to get the right detector. It has to be in the right place. It has to be of the right size. But the one question I want to speak about a bit this evening, or this afternoon, is, does this actually give the best optical sectioning in the easiest way? Well, we know that one way to look upon the optical sectioning is that the actual pinhole serves to obscure the light, and you only get a big signal from the light coming from the center. Absolutely true. And there are all kinds of formulae for what the strength of the optical sectioning is as a function of the pinhole size. You don't need to use a pinhole, and actually you can do, in some senses, better if you use a single mode optical fiber. You get exactly the same kind of optical sectioning. It gets more interesting. I'm not going to go into this in any great detail. If instead of using a fiber which supports just one mode, we have one which supports both the fundamental and the first order. Because if you do that, you find that you can form an image which is a combination, and you can separate out the various components of both a confocal image, a differential amplitude image, and a differential phase image, or any combination of the above. So one can actually be slightly more imaginative than a simple pinhole. But we all know what the problem is. The conventional microscope gives us this out of focus blur, which means we can't easily image specimens in 3 dimensions. So really the question is, we know that a microscope, if you like, is no good at this. Can we invent an optical system which will do what we want? So the idea is, can we form an optical system where the image on the right hand side is a scaled version in X, Y, and Z of the image on the left hand side? I'm not going to go into great detail on this, and the answer turns out to be you can. But the problem is, you have to build a system such that the angle with which a ray leaves the specimen, gamma 1, equals the angle with which it arrives in the image, gamma 2. If you do that, you find that the magnification is just the ratio of the refractive indices, N1 to N2. That means in air, you can get a perfect image, just what you want, with a magnification of 1. And if you use oil immersion, it goes up to 1.5. No one's going to be very interested in that. So you have to basically eliminate the out of focus blur so that one can, in essence, concentrate on very good 2-dimensional imaging, and then build up a through focus series to get the 3D rendering indirectly. So what's wrong with the confocal microscope? Well, for our purposes, it has to use a laser. For our purposes, let's imagine, because it makes the story more interesting, that they don't image usually in real time. You've got to put the pinhole in the right place, and you have to scan. So what would you like to do? Fairly obviously, you don't want a laser. You want it to be real time, easy to align, and no need to scan. Now, what have I just defined? I've defined a microscope with no laser, real time, no alignment, no scanning. That is what this thing does. So I've invented a microscope that Hooke had years and years and years ago, but it didn't have optical sectioning. So the question is, can we get optical sectioning into Hooke's old microscope? Now we know from our knowledge of confocal microscopes that the confocal microscope has a point illumination and a point detection, and it has optical sectioning. We know that the conventional microscope has uniform illumination, uniform detection, and does not have optical sectioning. So what do? Well, we ask Ernst Abbe. And we don't have to go very far, because Abbe wrote lots and lots of letters, and here he is explaining his theory of the resolution of the microscope. This is in Abbe's own handwriting, and if you go to Jena, you can see a statue here on which he has his D equals lambda over 2n sine alpha resolution thing. But he basically said, look upon the specimen as a diffraction grating. Think in terms of spatial frequencies. So let me say what this optical sectioning is in exactly the same way that I've done here, but in terms of spatial frequencies. The frequency of the structure of the illumination in a conventional microscope is 0. The frequency of the structure of illumination from a pinhole, let's say, is many, huge numbers in the confocal. Frequency of the structure of the detection is 0 in conventional, many in confocal. Confocal has sectioning, conventional does not. So the idea is 0 spatial frequency doesn't give you sectioning, all spatial frequencies do. It turns out that in the confocal illumination results in an averaging over all spatial frequencies. I'm not going to go into the mathematics. So if it's an average, it can't be the best. Now, an appropriately chosen spatial frequency, therefore, must be better. So the approach is to use a single spatial frequency carrier to ensure good sectioning, and then find a way to demodulate, or to put it in English, to remove the carrier. So this is the whole idea of structured illumination. You have a structure to cause the optical sectioning to happen, and then you have a demodulation phase to remove the structure. And this can be done easily as an add-on to a conventional microscope. Let me try and explain. If I try to image this part of a transistor here, if I project a pattern, a grid line onto the specimen, you can only see the grid line where the microscope is in focus. So what you have to do is invent a way to get rid of the bits of the specimen where there are no grid lines, and then get rid of the grid lines. And so what you basically have is a background image which you aren't interested in, together with a sectioned image modulated by these fringes. And one way you can get rid of them is computationally. You take, for instance, 3 different images where you slightly move the grid between each of them, and you computationally work out what's happening. And all you need to modify the microscope here is to add in a paddle. Here is the OptiGrid paddle to the illumination. And then you're able to turn conventional images like this into optically sectioned, crisper images like this one. You might think it's unnecessary to do the computation, and you have to take 3 images and you have to make sure they're the right 3. It turns out that instead of that, if you pass the composite image back through a reciprocal mask, exactly the same as the mask you used to illuminate, and you spin it, you, in essence, in a very hand-waving explanation, you perform synchronous demodulation, and you are able to extract the optically sectioned image. And if you do that, and if you get the frequency of the grid correctly, it's very light, efficient, and actually one can get enhanced optical sectioning. The blue line here is the ideal optimum spatial frequency for sectioning. The purple line is the ideal point scanning confocal, which of course doesn't exist. And the other one is the ideal slit detector. So you start off, you can get images like this as I focus through this specimen. They're optically sectioned, and you can add them all up to get an extended focus image. And as Pip was indicating earlier, these were taken with a light, with a candle, if you like, the ultimate laser-free confocal. So we started working on this, having had the idea. We then started to do some optical design. We then started to see how it might fit around a microscope, and a bit later on it looked a bit like this, and it now looks a bit like this. this is the clarity which people speak about more tomorrow, and it's an add-on to a conventional microscope, and it, like all these other things, turns images looking like this into images looking like that. It can also get things moving quite quickly. So here is a zebrafish with its heart beating. Now, the other thing the clarity can do, apart from the conventional, it can take the confocal. So what was being shown to start off with was the confocal image, and then switching to the conventional a bit later on. More of these videos, because they're quite pretty. This is a brassica thing. You can get these really rather nice three-dimensional images, and more and more. I just show them because these instruments really form images which look exactly like, if you like, a real confocal microscope, because it is a real confocal. It can be used in other applications. Here's the 3D histech slide scanner, where the normal fluorescent image can be improved to making it confocal. I started off by saying one of the applications was metrology, and here it is in the guise of the Zeiss SmartProof 5. And here is a thing that is trying to be profiled. It's a dental implant, and the interest is in the angle of the implant, which is supposed to be 120 degrees. Pip mentioned the Unity, the laser-free confocal, so I shan't speak about that anymore, other than to say here is a scientist in a white coat using it, and the whole thing is controlled, as she said, from this tablet. So, new microscopes for all. You can do an enormous amount with Hooke's microscope. You can probably do a little bit more with either Clarity or Unity, but they both produce images which are substantially the same as you would get with a traditional confocal, but hopefully at a lower price, and I have no idea what the prices of these things are. And in this instance here, you just put in the specimen and press go. Okay, I think that's all I want to say, Rimas.
Thank you, Tony. Thanks. Entertaining as always. We have one sort of quick question on our chat line. Jan Pechle is asking, can you give your personal perspective on Petrans and Hadravsky's work?
Yes.
Early work on a Nipkow disc confocal?
Yes, I actually had a lecture on this. I have nothing but the highest regard for Petran and Hadravsky. Actually, what Petran did was really quite remarkable. When he first built his microscope, the question is, what did he use for a light source? Because quite clearly there was no no laser or anything. And
a candle, perchance?
No, well, sort of. A rather well-known candle called the Moon, or rather called the Sun. He took the microscope up into the High Tatras Mountains and used the Sun as the light source. But I've been very fortunate to know Moimir and Milan for the last, I don't know, 30 or more years. Milan, of course, died a few years ago. And I think their work, and particularly where they were doing their work, is extraordinarily impressive. Both, I might say, not physicists, but medical doctors.
Okay. Kirti Prakash is asking, why didn't Confocal get a Nobel Prize?
Well, I wasn't on the committee, so I can't answer that.
He also says here, he heard that Nobel Committee didn't like outspoken Minsky. Didn't Minsky effectively drop this as soon as he invented?
Minsky, I don't think, I went to his house once, and I think he claimed to have built something which he couldn't find. I believe that to be true. But it wasn't what he was particularly interested in. Minsky certainly is outspoken, and he's not everyone's cup of tea, but I can't believe that was the reason.
Okay. I have a sort of question. I remember when I first came to this field, OBIC was still going strong and was regarded as a sort of a future of scanning optical microscopy. What became of OBIC?
I don't know. I think it was a very good technique. I mean, for people who don't know, it's the optical version of the electron microscope EBIC technique, where you scan an optical beam across a semiconductor, generates electron hole pairs which can probe various properties of the device. And in a way, it's superior to the electron beam because there's no electron beam damage. It didn't catch on in the way in which the acoustic microscope didn't catch on. I mean, really, all the optical microscope can do is measure refractive index. Well, frankly, who cares? The acoustic microscope, on the other hand, does measure serious material properties. So
we have a question here. So who was the person on the photograph?
Ah, Harry Nyquist.
Very good.
It's a great shame people that aren't remembered in this way, I think.
So we have one more question here. Is it possible to identify who actually built the first scanning microscope with a galvo mirrors?
Whew. I don't know that. I can identify that we built things with galvo mirrors that didn't work, but that probably isn't the
Fred Brackenhoff?
Could be, but he was doing stage scanning like us, as was Finance at Emble. So it was probably only when it started to become commercial that people started to do beam scanning. I mean, to be honest, we did stage scanning because it made the optics easier. You know, to build a proper beam scanner. Oh, wait one moment. The other people who built beam scanners very early on was the Japanese company Lasertech. And they had a hybrid scheme. They had a mirror for scanning in the relatively slow frame direction, and they had an AOD for scanning in the line direction, and that was a real-time microscope. So that was, I'm going to guess that might have been the first commercial high-speed beam scanner. But I don't really know the answer.
Okay, we have a question here that I can probably answer. What is the SOM? Scanning optical microscope. It's one of the older ways of calling it, I guess.
Yes, you see, when we started, we always called it a scanning optical microscope because that was what, I mean, it was a flying spot microscope, and the word confocal didn't come until later. Who coined it for the first time, I don't know. It could have been Fred Brackenhoff, as someone suggests. I really do not know. I mean, it's a thing we in Oxford used. We came up with the confocal, if you like, because we like mathematics. And if you stick a pinhole in the equation, it becomes a delta function, and the integral is easy. So we actually had a thing called a type 1 microscope, which was the regular confocal, and a thing called a type 2, which was the scanning microscope with the pinhole, which of course is the confocal. But who came up with it, I don't honestly know.
I think you really need to look for a laser physicist here because of confocal resonators.
Well, this is the whole thing. If you know a confocal resonator, you wouldn't call it a confocal microscope, you call it a concentric microscope because it's not a confocal. But I think we're being a bit too anarchy about this. It could be Kjell Carlson, it could be Niels Aslund. I wouldn't know. They were amongst the earlier people, and Niels Aslund had a company, the name of which I can't remember, which was selling beam scanners early on. You could be right. I really do not know.
Okay, guys. So if there are no more questions, I think we'll probably draw a line here.
Okay.
Thank you, Tony. Thanks.
Okay,
thanks again for a very sort of
slightly more, not quite as smooth as it hoped to be.
No, thank you very much,
Smooth
Tony.
enough.
I think there was something for everyone there from all different countries involved in the development as well. It's great, really, really good. Even I understood it. Said something.
Okay, yeah.
I mean, thank you. Actually, I'd like to take this opportunity to say thank you to everybody today, both for attending and for the questions, but obviously, in particular, to all the speakers who've done a great job in preparing and done a brilliant job at speaking to the wall in their spare room and listening to the quiet feedback. And I think everybody, by the looks of it, agrees. It's been a great day, and I think we're probably all worn out now. So please come back tomorrow. Join us in the UK at 9 o'clock in the morning, UK time. For the next session, which is on, I think, image analysis and AI, followed by adaptive optics, I think. And hopefully we'll see you all again in the morning. And thank you once again, Tony, for a great talk. And
Thank you.
Really nice to see where everything came from. Thank you, everybody.